Okay, welcome to Unit 4. So this unit corresponds to homework number 4, which you'll be ready to do after you watch this set of slides, and also to Chapter 9 in the textbook, which is all about microbial genetics. So we're first going to familiarize ourselves with some genetics vocabulary. So on this slide, we have a figure that shows a bacteria cell. It has a lot of information about the exterior layers like the capsule, the polysaccharide, and then it has some details on the interior of the bacteria. So all of the interior is shaded in gray, which would represent like the cytoplasm. And we have a black squiggly line in the cytoplasm, which is supposed to be a molecule of DNA, and it is the chromosomal DNA or the genome. So the chromosomal DNA, aka the genome, contains all the genes that are essential for the bacteria to live. We also have this small gray circle, which is representing a smaller piece of plasmid DNA. And plasmid DNA is not essential. It has genes that are going to be giving the bacteria special skills. It's much smaller than the chromosomal DNA or the genome, and it can be passed between bacteria pretty easily, as we'll learn in Chapter 9. So if we look at the chromosomal DNA and the plasmid DNA, this collectively is the cell's genotype. So it would be the sequence of nucleotides in these DNA molecules, and this sequence would be unique in each cell, and that sequence is what the genotype of the cell would be. The information encoded in the chromosomal DNA, the genome, or in the plasmid DNA, the non-essential genes, is going to be used by the cell to do certain jobs. And this physical expression of the DNA into protein is what is known as the phenotype. So in this particular cell, for example, it has a gene for how to make a capsule layer. And when that gene is turned into RNA and then into protein, the protein will physically make the capsule, and this will give the bacteria the phenotype of having a capsule. So genotype is the information encoded in the DNA, and phenotype is a physical expression of that information. So we've just described the process of turning DNA into RNA by transcription, and then turning that RNA into protein by translation, where the DNA has information and then the protein carries out those instructions and actually does a job for the cell, like building a capsule. Another thing that cells will do with DNA is they will replicate it, which is simply making a copy, and this will be done in preparation for cell division. So bacteria cells will do these processes, as will human cells. So all living cells are going to do replication, transcription, and translation with their DNA molecule as a starting point. So shown in figure 9.6 from your textbook is a picture of a DNA molecule being replicated. So first, a few points about a molecule of DNA. In this image, I would like you to notice the top portion of figure 9.6, where we have two ribbons swirling around each other in a helical shape to represent the DNA molecule. So a DNA molecule is made up of two strands, so each of these ribbons is one of the strands of DNA. The interior of the molecule has nucleotide bases, either adenine, cytosine, guanine, or thymine, which are usually abbreviated A, C, G, and T. And these nucleotide bases of one strand will be complementary to a nucleotide base on the other strand. So a nucleotide base of strand 1 will bind to a nucleotide base on strand 2 of the same DNA molecule. And this complementary binding is always between A and T, or adenine and thymine, and then always between C and G, or cytosine and guanine. So when the DNA molecule is replicated, the double helical shape opens up and each of the strands are separated into the structure at the bottom of figure 9.6, which is called the replication fork. So when each of these two strands is exposed, enzymes can go into the interior of the replication fork and can start synthesizing a new strand of DNA that is complementary to the old one. So in this figure, we have the old strands of DNA from the original DNA molecule in blue and the new strands being synthesized in red. So for this figure, I'd like you to be familiar with all of the enzymes listed on this figure, and each of the enzymes is going to end in an ASE. So we have topoisomerase, helicase, DNA polymerase 3, RNA primase, and DNA ligase. So you should be familiar with the functioning of those enzymes in DNA replication. So after these enzymes have done their work, there will be two molecules of DNA, each made up of two strands of DNA. So we have one molecule which will finally be formed on the left of the fork, having a blue strand and a red strand that will wind up into a double helix. And then we'll have the second molecule of DNA, which is on the right of the replication fork, made up of a blue strand and a red strand that will then wind up into a double helix. And each of these two DNA molecules will then be divided into the daughter cells. So the original parent cell will divide into two, and each of the daughter cells has a molecule of DNA that was identical to the parent cell molecule of DNA, thanks to DNA replication. So what we've just described is bacteria passing on genetic traits or genetic information to offspring in vertical gene transfer. So humans also do vertical gene transfer. From parent to offspring, DNA is passed to the next generation, and the parent and daughter cells are related, and this would be a vertical transfer of information. Bacteria have the unique ability of doing additionally horizontal gene transfer. And there are three types of horizontal gene transfer outlined in Table 9.3. So horizontal gene transfer is the sharing of genetic information, or DNA, between cells that are in the same generation and that are not related to one another. So these could be between bacteria of the same species, although not related. They could be related, or they could be between bacteria of even different species. So we'll go through the three types of horizontal gene transfer, starting with conjugation. So conjugation is a type of horizontal gene transfer where unrelated bacteria can share DNA with each other. So there's a donor that provides a piece of DNA to a recipient. And in conjugation, we have to have a physical connection between the donor and the recipient through a structure called the pilus that some bacteria are able to extend and connect with a recipient bacteria. So this image shows three bacteria cells drawn in a very simplified way. The genome, or the bacterial chromosome, is shown in blue, and the plasmid, if it is present, is shown in pink. So the first cell in the middle we'll focus on is called an F- cell. So you'll notice this cell has a bacterial chromosome with all essential genes, but it does not have the additional plasmid. So this is described as an F- cell. In a conjugation, this F- cell can only be a recipient of transferred DNA. On the left, we have an F+ cell. So this cell looks identical to the F- cell with the addition of an F plasmid, which has genes for extending a pilus and connecting with a recipient. So this F+ cell, because it has that genetic information, can extend pili and donate DNA to a recipient through horizontal gene transfer by conjugation. The cell on the right is an HFR cell. So what's happened in this bacteria cell is the F plasmid and the bacterial chromosome have recombined into one large piece. Because this HFR cell has the F plasmid, it has the genes for information on extending a pilus and connecting with the recipient. So the HFR cell also can be a donor in horizontal gene transfer. So we have shown in figure 920 two different conjugations. So in the top, we have a donor cell is the F+ cell, so it has its genome or chromosomal DNA in blue, and the F plasmid is shown in a multicolor ring. The recipient is an F- cell, which has its chromosomal or genomic DNA in purple. So because the donor F plus cell has information for extending a pilus, it extends the pilus and connects with the recipient. And this forms a tunnel between the two cells. That DNA is shared from the donor to the recipient. So the plasmid is replicated. We have now 2 copies, and one of the copies is moved into the recipient cell. And so at the end of the conjugation, the donor is still an F plus cell, but the recipient, which has now gotten the F factor, has become an F plus cell, where it was originally an F minus cell. The other conjugation shown at the bottom of figure 920 is between an HFR cell as the donor and an F minus cell as the recipient. So the HFR cell has its genomic DNA colored in blue, and the F factor is incorporated into the genomic DNA. When the donor cell extends a pilus using instructions in its F factor genes, it connects with the recipient, replicates a portion of its DNA, and passes that DNA into the recipient. So in the HFR transfer, the F factor DNA is transferred, and some of the genomic DNA is also transferred. So some blue DNA gets into the recipient as well. So at the end of the transfer, the recipient DNA has gone from being an F minus cell to being an HFR cell. So it's gotten F factor and some additional genomic DNA from the donor. The second type of horizontal gene transfer is transformation. Unlike conjugation, this does not require a physical connection between the cells. In this figure, we have the cell on the left with a broken plasma membrane and cell wall is the donor. So this cell is damaged and will die, and so is releasing its DNA into the extracellular environment. Its DNA is colored in blue and is chopped into small pieces as the cell breaks down. The recipient cell is the cell in the middle, which encounters a naked molecule of DNA, meaning the DNA is outside of a cell, takes it up into its cytoplasm, and then that new piece of DNA recombines in the recipient, and the new DNA and the original genomic DNA in purple become one piece. One note about the recipient and transformation: this cell has to be competent, meaning it has to be able to take DNA up from the outside environment into its interior. So some bacteria cells are competent and can accept DNA by transformation. Some bacteria cells are not. There was a very famous experiment performed by a scientist named Griffith, and it was demonstrating the concept of transformation. So I'll walk you through that experiment, which is illustrated nicely in figure 922. So what Griffith did is he used 2 different types of bacteria. He had one type, which he called the S strain for smooth, because they had a capsule around their cell membrane and cell wall. They also, when growing on a petri dish, had a very smooth appearance of the colony. The other strain was the same species of bacteria, but he called them R strain because they had no capsule, and when they grow on a petri dish, the appearance of the colony is very rough and bumpy. When Griffith injected the S strain into mice, they cause a disease that is very severe and causes the mouse to die. When Griffith injected the R strain into mice, the mice were able to survive. The reason for the difference in the effect on mice is because of the capsule. When the S strain are injected into the mouse's immune system is not able to clear them, where when the R strain is injected into the mouse's immune system is able to clear the bacteria and destroy it, so the mouse is not affected. And I want to bring your attention back to the benefit of having a capsule for a bacteria. So the phagocyte is one of the first responders to infection, and bacteria having a capsule are not able to be destroyed by a phagocyte. So for this reason, the S strain was able to escape the phagocyte in the mouse and then went on to increase in numbers and cause an infection, where the R strain was attacked by the phagocyte and destroyed, and the mouse was fine. Okay, so an additional condition in Griffith's experiment was he took S strain, which normally would kill the mice, heated up the bacteria so they were killed, and injected the heat-killed S strain into a mouse, and the mouse survived. In condition D, Griffith mixed heat-killed S strain with live R strain and injected this combination of the two cells into the mouse and found that the mouse dies. The reason the mouse dies in condition D is that the heat-killed S strain releases its DNA when it's heated. That DNA is accepted by the competent R strain by transformation. Some of the R strain are converted to live S strain and are then able to cause infection in the mouse and lead to the mouse's death. So this experiment by Griffith was proof that transformation can occur, that the R strain in his experiment were transformed into S strain by naked DNA released from heat-killed S strain. The last type of horizontal gene transfer is transduction. And in this type, we have a bacteriophage, which you may recall is a virus that infects bacteria cells, that infects a bacteria cell, as shown in the top of figure 9.28, and injects its viral DNA or phage DNA into the interior of the bacteria cell. The next step is that the viral nucleic acid or viral DNA takes over the host bacteria cell so that the host cell becomes a factory for viruses, and all of its cellular machinery are put towards making new viruses, with one small error. So you'll notice in the third cell down, we have 4 bacteriophage in the interior of the bacteria cell. 3 of those bacteriophage have viral DNA in their capsid, which is red. 1 of the bacteriophage incorrectly has bacterial DNA inserted into its capsid, which is drawn in pink. When these phage lyse the bacteria cell, the viral DNA containing bacteriophage will go on and cause further viral infections of other bacteria cells. But the phage with bacterial DNA in its capsid will transfer bacterial DNA from the donor, which was the originally infected bacteria cell, to a new recipient. So when this phage injects DNA into the interior of the bacteria, it inadvertently and unknowingly injects bacterial DNA, which is then recombined with the bacterial chromosome and incorporated, as shown in the very bottom cell, with the genomic DNA being purple and the newly inserted bacterial DNA from the donor in pink. So the donor and recipient are never in direct contact in transduction. They are just sharing DNA through a virus mediator. Okay, the last subject in Chapter 9 is DNA mutation. So DNA mutation would be a change in DNA sequence. And this can result in a change for the cell and the cell's phenotype, or it may not result in a change for the cell and the cell's phenotype. So we're going to discuss an experiment that is frequently done to identify chemicals that cause DNA mutations. And these chemicals are classified as mutagens. They have the ability to alter DNA sequence. So this test is the Ames test, and it's outlined in figure 927. So we mix first in a test tube a culture of Salmonella bacteria. These bacteria are auxotrophic, which is a vocabulary word you can look up. And auxotrophic means they're unable to make something essential. In this case, they're unable to make the amino acid histidine. So they're described as histidine minus. We also, in this culture tube, would mix liver enzymes, which will take the chemical we're testing, break it down into metabolites. So we're testing the chemical and all of its metabolites for mutagenic properties. When we plate some of this Salmonella bacteria on agar that does not contain histidine, these bacteria should not be able to grow. They are only able to grow if we provide histidine for them. And since we are not, they should not be able to grow. So the control plate is made up of media lacking histidine and also lacking a test chemical. And we see after plating these salmonella bacteria, a few colonies arise by spontaneous or random mutation. They've become histidine plus, are able to make their own histidine, and are able to grow on the orange control plate. We also plate a portion of the culture, the liquid culture, onto the test plate, which is identical to the control plate with the addition of the test chemical, which we're trying to determine if it is a mutagen. So on the test plate, we see growth of many more colonies. So many more bacteria have gone from being histidine minus or histidine oxytrophic to histidine plus and now growing on this media without histidine provided. Because we have so many more colonies on the test plate than the control plate, we can conclude that this test chemical, or one of its metabolites, is a mutagen and is causing DNA changes and increasing the rate at which histidine minus cells become histidine plus cells. With that, we conclude Chapter 9. So good luck on your handout for this lecture slide and your homework.